Somewhere beyond the edge of a tropical beach, where turquoise water fades into dark blue, the ocean floor simply disappears. A diver descending alongside an island wall in the Bahamas can move through warm, clear water filled with reef fish, look over the edge, and find nothing beneath them but thousands of meters of open water. The island ends. The abyss begins. Most people who visit tropical islands never realize they are standing within swimming distance of one of the most extreme environments on Earth. The deep sea gets all the fear, the trenches, the crushing pressure, the darkness, but the deep sea is isolated. Island walls are not. They are places where the full depth of the ocean rises startlingly close to the surface. The Cayman Wall drops from shallow reef to more than 6,000 meters deep within a remarkably short distance. Creatures that spend their days hundreds of meters below the surface climb these walls every night, reaching depths used by recreational divers before vanishing again by dawn. Deep-sea sharks have been filmed near island walls at depths where they should not be. Coral colonies growing on these cliffs have been alive for thousands of years, and every night, billions of animals move up from the darkness along these underwater walls in one of the largest migrations on Earth. What comes up those walls after dark? What lives permanently on the cliff face itself? And why do marine biologists who study both environments often describe island walls as less predictable than the deep sea? By the end of this video, you'll understand why the ocean beneath islands creates one of the strangest collision zones on Earth, where shallow reefs, abyssal depths, ancient life, and deep-sea predators all meet in the same column of water. Seen from above, an island appears self-contained, a small piece of land surrounded by an immense circle of water. Maps reinforce the illusion. Shorelines seem to define the island's boundaries, as though the visible land is the whole structure. In reality, the land above the surface is only the smallest fraction of something far larger. Most oceanic islands are mountains, but unlike the mountains of continents, the overwhelming majority of their mass remains hidden beneath the sea. Their story begins deep below the ocean floor, where magma rises through fractures in Earth's crust and slowly accumulates layer upon layer. Across immense spans of time, volcanic rock builds upward from abyssal depths until the summit finally breaks the surface. What emerges as an island is merely the highest point of a geological structure that may extend several kilometers downward into darkness. The beaches, forests, cliffs, and reefs visible from shore occupy only the uppermost tip of a submerged mountain, whose foundations disappear into the deep ocean basin. Many tropical islands later acquire another layer of life. Coral colonies settle around the volcanic base and begin constructing reefs of limestone. As the underlying volcano gradually subsides, the coral continues growing upward towards sunlight, maintaining its position near the surface. Over countless generations, this process can create vast reef rims and circular atolls surrounding tranquil lagoons. Yet beyond the outer edge of those reefs, the architecture changes abruptly. The living platform ends. The open ocean begins. This boundary is where the island wall emerges. It is rarely a gentle descent. More often, it resembles the side of a submerged cliff, a steep face plunging downward through successive layers of the ocean. In some places, the drop approaches true verticality. A diver drifting beyond the reef edge may find the seafloor vanishing almost instantly, replaced by a descending wall that continues far beyond the limits of visibility. The wall itself is not smooth. Ancient volcanic eruptions left terraces, ridges, chimneys, and fractured ledges throughout the rock. Later, currents carved channels through weaker sections, while chemical erosion widened cracks into crevices and caves. Living organisms added further complexity. building coral structures, skeletal frameworks, and dense biological growth upon the stone. Over millions of years, the result became something closer to a vast underwater landscape than a simple cliff face. These formations occur throughout the world, wherever steep volcanic islands rise from deep ocean basins, across the Caribbean, through the island chains of the Pacific, and throughout the Indo-Pacific. The same pattern appears again and again. A narrow ring of shallow water gives way to an immense descent. The geography may differ in detail, yet the underlying structure remains remarkably consistent. What makes island walls extraordinary is not simply their depth, but their proximity. The average depth of the global ocean approaches 4,000 meters. Along continental margins, reaching such depths often requires traveling enormous distances offshore across broad continental shelves. The transition is gradual, almost imperceptible. Around oceanic islands, that gradual transition is absent. Deep water arrives almost immediately. A person standing on a beach may be only a short swim from an environment that extends thousands of meters downward. Within a distance that seems insignificant on a map, warm sunlit shallows connect directly to the vast interior of the ocean. The wall forms the bridge between those worlds, compressing an immense vertical range into a remarkably small horizontal space. Everything that makes island walls unique begins with that simple piece of geometry, the deep ocean rising astonishingly close to the surface, close enough that two realms which normally remain separated by distance are brought into quiet and continuous contact. Far from any shoreline, the ocean organizes itself into layers, not visible layers like bands of color, but living layers, each occupied by creatures shaped by a particular depth, pressure, temperature, and rhythm of light. Many species spend their entire existence within a remarkably narrow vertical range. Their lives unfold above invisible boundaries or below them, and for millions of years those boundaries have acted almost like borders between separate worlds. A fish adapted to bright surface waters rarely descends into darkness. A predator built for the deep seldom rises into the illuminated upper ocean. Evolution has encouraged specialization rather than travel. Over immense stretches of time, entire communities have formed in relative separation, each responding to environmental conditions that neighboring communities may never experience. The result is a vertically structured ocean, a living architecture extending from the surface toward the abyss. Island walls interrupt this arrangement in a subtle but profound way. The stone face descending through the water column creates a continuous pathway connecting environments that are normally divided by great distances. Instead of existing as isolated layers suspended in open water, species from different depth zones find themselves linked by the same geological structure. The wall becomes a shared boundary where separate ecological worlds brush against one another. Marine biologists sometimes describe these regions as biological collision zones. The phrase does not suggest violence so much as proximity. Organisms that evolved under entirely different conditions suddenly occupy neighboring space. Along the steep walls surrounding deep basins, such as the tongue of the ocean in the Bahamas, reef fish move through water that may also contain midwater hunters and visitors from far below. Within a surprisingly narrow column of ocean, communities that would normally be separated by hundreds or even thousands of meters become compressed together. This compression creates encounters that are rare elsewhere in the sea. A predator accustomed to hunting among coral formations may drift only a short distance from an animal whose ancestors spent millions of years in perpetual darkness. Neither lineage evolved with the other in mind. Their sensory systems, behaviors, and survival strategies developed under different rules. Yet along the wall, those rules overlap, producing relationships that have no true equivalent in the open ocean. The consequences extend beyond feeding. Every organism releases traces of itself into the surrounding water. Chemical compounds disperse in slow currents, carrying information about territory, reproduction, stress, and the presence of prey. Along island walls, these signals intermingle across depth zones. Scents originating from deep environments move upward. Signals produced near the surface drift downward. Species encounter information that would rarely reach them elsewhere, creating a chemical landscape of unusual complexity. Sound follows a similar path. Vibrations generated by movement, feeding, communication, and navigation travel through the water and interact with the contours of the wall. Pressure waves originating in one depth zone propagate into another. Creatures adapted to one acoustic environment find themselves immersed in echoes and frequencies arriving from unfamiliar biological communities. The wall gathers these signals and concentrates them into a shared corridor. Light contributes its own layer to the exchange. Bioluminescent organisms ascending from darker regions carry flashes and pulses into waters where daylight species dominate. These displays evolved for communication, camouflage, courtship, and defense within deep habitats. Near the wall, however, their signals may appear before animals that possess no evolutionary history of interpreting them. Ancient conversations from the deep briefly enter another realm entirely. Among the most striking examples are six-gill sharks, typically associated with depths far below the reach of ordinary reef life. They have been documented ascending island walls around Bermuda and Fiji until they occupy waters used by shallow species. The distance separating these communities is not measured across vast horizontal expanses, but through a relatively small vertical interval. Two branches of ocean life, long divided by depth, momentarily inhabit the same neighborhood. The wall therefore becomes more than geology. It becomes a place where the ocean's separate layers lose some of their isolation, where boundaries soften, and where life from different worlds shares the same descending face of stone in quiet and continual contact. As daylight fades across the ocean, a vast movement begins. There is no signal visible from above, no dramatic shift on the surface, no indication that anything unusual is unfolding beneath the waves. Yet through the darkness, countless living creatures begin to rise. It is one of the largest daily migrations on Earth, a phenomenon so immense that its total mass is measured not in thousands or millions of tons, but in billions. Marine scientists call it diel vertical migration. Every night, organisms that spend the day in deeper water ascend toward the surface under the cover of darkness, then retreat again with the return of morning light. This rhythm repeats every day of the year across nearly every ocean basin on the planet. Vast populations of small animals, fish, crustaceans, and drifting predators participate in a synchronized ascent that has likely been occurring for tens of millions of years. In the open ocean, this movement unfolds across a huge vertical expanse. Creatures may travel hundreds of meters upward through featureless water before reaching feeding grounds closer to the surface. Around island walls, however, the journey takes on a different character. The migration becomes compressed against the descending face of the island. Instead of crossing empty space, countless organisms move along a physical structure that guides their path through the water column. The wall provides orientation in an environment where few landmarks exist. Its ledges, crevices, and contours create a vertical corridor stretching from shallow water toward the depths below. As darkness settles, streams of life begin moving upward along this submerged landscape. What appears quiet during daylight becomes animated by continuous motion. Lanternfish emerge first in many locations, their bodies carrying tiny light-producing organs that shimmer faintly in the darkness. Hatchetfish follow, silver and delicate, built for life in dim water. Bristlemouth fish, among the most abundant vertebrates on Earth, join the ascent in numbers almost beyond imagination. Deep water shrimp rise with them, along with gelatinous siphonophores that drift like living chains suspended in the current. Together they form a moving river of biomass flowing upward through the night. Predators follow close behind. Many spend their days several hundred meters below, conserving energy in colder, darker water. After sunset, they ascend along the wall, tracking the migration. Species that may remain hidden at depths between 400 and 800 meters during daylight can arrive astonishingly close to the surface before dawn. The vertical separation between deep and shallow ecosystems narrows dramatically for a few brief hours. Researchers studying island walls have documented species appearing at depths around 30 meters, despite being previously associated with habitats hundreds of meters deeper. The wall effectively compresses their ecological range. What would normally be separated by vast stretches of open water becomes concentrated into a relatively narrow corridor. Animals adapted to very different environments find themselves moving through the same nighttime pathways. For those who enter these waters after dark, the transformation can seem almost unreal. Bioluminescent organisms drift through the water column in luminous clouds. Tiny disturbances trigger flashes of blue and green light that vanish as quickly as they appear. Unfamiliar silhouettes emerge at the edge of visibility and disappear again into darkness. The sense of vertical space changes as the migration unfolds, the water filled with travelers moving between worlds that rarely overlap during the day. Then, almost as quietly as it began, the movement reverses. The first hints of approaching dawn send the ascending communities downward once more. Lanternfish descend, shrimp retreat, predators follow. By sunrise, the wall appears transformed again, inhabited largely by its daytime residents. The visitors from deeper water have withdrawn into darkness, leaving little trace of the immense passage that unfolded only hours earlier. The stone face remains, silent and still, while another cycle completes itself within the ancient rhythm of the ocean. Some inhabitants of island walls never participate in the great nightly ascent, while entire populations move through the water column according to the rhythm of darkness and dawn. Other organisms remain attached to the wall for decades, centuries, and sometimes millennia. They do not travel between worlds. They belong entirely to the vertical landscape itself, shaped by conditions found nowhere else in the ocean. These permanent residents occupy a habitat that is neither reef nor deep sea. The wall creates its own environmental identity, a narrow but remarkably stable zone where geology, current, depth, and biology combine into something distinct. Marine researchers often describe these assemblages as wall-specific communities, collections of species whose lives are tied not simply to depth, but to the physical presence of the wall itself. Remove the wall, and the community disappears with it. Among the most remarkable of these residents are the organisms known as black corals. Despite their name, living colonies are often pale gold, ivory, or luminous white. The dark color appears only after death, when the organic skeleton beneath the living tissue becomes exposed. These corals establish themselves along deeper portions of island walls, generally below the brightest waters, where sunlight weakens and the surrounding environment settles into a quieter rhythm. Growth occurs with extraordinary patience. Year after year, branch after branch extends into the current. Some Caribbean colonies have been dated to more than 2,000 years of age. Long before many events recorded in human history, these organisms were already anchored to the same sections of wall, filtering food from passing water and adding another layer to the living architecture around them. Their existence stretches across timescales rarely encountered in the visible world. Nearby, deep-water Gorgonian fans spread outward like vast underwater canopies. Some reach widths of several meters, forming delicate frameworks suspended from the rock face. At the depths where many of these colonies thrive, photosynthesis no longer provides energy. Instead, they rely upon a constant stream of microscopic particles carried by the surrounding water. Each branch acts as a living net, capturing nourishment delivered from distant places throughout the ocean. The wall plays a crucial role in sustaining this abundance. Ocean currents moving across open water eventually encounter the vertical face and are forced to change direction. Some portions of the flow are diverted upward, others are guided downward. The result is a continuous redistribution of water that concentrates nutrients along the wall. Tiny organisms, drifting organic material, and suspended particles become trapped within these pathways, creating an unusually reliable food supply. This process transforms the wall into a surprisingly rich environment, despite its location within ocean regions that may appear relatively barren at the surface. Vast stretches of tropical ocean are often nutrient poor, their clear blue waters reflecting a scarcity of suspended life. Yet along the wall, invisible deliveries arrive continuously. Food passes by every hour of the day, carried not by chance, but by the geometry of the landscape itself. As a result, life accumulates. Sponges attach themselves to overhangs. Filter feeders crowd narrow ledges. Small invertebrates occupy cracks too narrow for larger animals to enter. Countless species layer themselves across the rock face, each finding a niche within the complex structure. In certain locations, surveys have recorded densities of living biomass that exceed those found on nearby reef flats. The vertical surface supports an astonishing concentration of life. What emerges is not a border separating two ecosystems, but a complete ecosystem in its own right. The wall does not merely connect shallow and deep environments. It generates conditions neither of those environments can produce independently. Through the steady delivery of nutrients, the persistence of ancient organisms, and the intricate architecture created over immense spans of time, the wall becomes a place with its own biological identity, enduring quietly along the submerged slopes of islands across the world. Water appears effortless when viewed from above. A calm ocean surface can suggest stillness, as though the sea simply rests around an island and changes only when storms arrive. Beneath that appearance, however, enormous rivers are already in motion. They have no banks and no visible edges, yet they travel across entire ocean basins, carrying heat, nutrients, oxygen, larvae, and microscopic life through a constantly shifting network of currents. When these moving waters encounter an island, they meet an obstacle that cannot be crossed. The island interrupts flows that may have traveled thousands of kilometers uninterrupted. Water is forced to respond. Some streams bend around the island's flanks, others are pushed upward, others descend. The result is not a single current, but an intricate architecture of movement surrounding the submerged mountain. Along the wall itself, this architecture becomes especially complex. Deep water moving through the ocean occasionally collides with the steep underwater slopes and is redirected toward shallower depths. Oceanographers refer to this process as upwelling. Water that may have spent years or even decades in darkness begins a rapid ascent. Cold layers, rich in dissolved nutrients, rise from depths of hundreds, sometimes thousands, of meters and enter parts of the ocean normally influenced only by surface conditions. These nutrients originate from a long journey through the deep sea. Organic material slowly sinks through the water column, and decomposes, releasing compounds that accumulate far below the reach of sunlight. In much of the open ocean, these nutrients remain trapped at depth, unavailable to surface ecosystems. Around island walls, the geometry of the landscape changes that pattern. The upward movement of deep water delivers this hidden reservoir back toward illuminated layers, where life can immediately make use of it. The biological consequences are profound. Microscopic phytoplankton flourish when nutrients arrive. Tiny grazers feed upon the blooms. Small fish gather where food becomes abundant. Larger predators follow them. Energy that would otherwise remain dispersed across a vast volume of water becomes concentrated around the island. The remarkable productivity observed around many island walls begins with this continuous lifting of deep ocean chemistry toward the surface. Yet the movement is not entirely upward. On other sides of the island, surface water can be forced downward. This process, known as downwelling, carries oxygen-rich water and organic material into deeper environments. The descent replenishes habitats that would otherwise receive fewer resources from above. While one current brings the deep ocean upward, another returns elements of the surface world downward, creating an ongoing exchange between layers that are often separated elsewhere. Together, these movements generate what oceanographers call the island mass effect. The phrase describes a measurable increase in biological productivity surrounding oceanic islands compared with the open ocean around them. From satellites observing chlorophyll concentrations to instruments measuring nutrient levels below the surface, the signature appears repeatedly. Islands influence the water around them far beyond the visible shoreline. The complexity continues downstream. As currents flow past an island, they often shed rotating bodies of water known as eddies. These slow-spinning vortices can persist for days or even weeks, gathering plankton, nutrients, fish larvae, and drifting organisms into concentrated pockets. Rather than dispersing across the ocean, resources remain trapped within the rotating circulation. Many marine animals learn these locations with remarkable consistency. Predators return repeatedly to productive eddies, finding prey concentrated within a relatively small area. What appears from above as ordinary ocean may contain invisible structures shaping the movements of entire food webs. Along certain sections of island walls, current speeds become surprisingly powerful. Measurements around the Cayman walls have recorded flows exceeding 2 knots, strong enough that unaided swimming against them becomes impossible. Yet these conditions do not remain fixed. Seasonal shifts alter circulation patterns. Weather systems reshape local flows. Tides introduce additional variation. The arrangement of water around the wall is constantly being rewritten. Because the currents themselves are always changing, the living community responds in turn. Nutrients arrive from different directions. Feeding opportunities expand and contract. Species gather, disperse, and return. The wall remains anchored to the island, but the moving architecture surrounding it is never entirely the same from one day to the next, quietly reshaping life through the language of water in motion. Unlike light, nutrients, or temperature, pressure is never seen. It leaves no color in the water and no visible boundary across the sea. Yet it is one of the most powerful forces shaping life in the ocean. From the moment a creature begins descending below the surface, the weight of the water above starts to increase. Every additional 10 meters adds another atmosphere of pressure, an invisible burden that grows steadily with depth until it reaches values almost impossible to imagine from the surface world. For most forms of life, this pressure acts as a barrier. Creatures adapted to shallow water carry bodies designed for relatively stable conditions. Their tissues, organs, and biochemical processes function within a narrow range. Far below them, deep-sea animals have evolved under circumstances so different that the two groups often remain permanently separated. Pressure helps maintain this divide. It is one of the reasons the ocean can contain multiple biological worlds, stacked vertically atop one another. Island walls compress that separation into a surprisingly small space. The transition from surface conditions to extreme pressure occurs not across vast horizontal distances, but along a steep descent beside the island itself. This creates an unusual setting where animals capable of crossing those boundaries can move directly between environments that would otherwise remain isolated. Among the most remarkable of these travelers are sperm whales. Their immense bodies appear perfectly suited to the open ocean, yet they repeatedly descend alongside island walls into depths exceeding 1,000 meters. The wall provides a reliable geographical reference through an environment where visual landmarks rapidly disappear. Following these submerged slopes, sperm whales leave the sunlit ocean behind and enter a realm where pressure climbs to levels that would be lethal to humans within moments. Their survival depends on a suite of extraordinary adaptations refined over millions of years. During descent, their lungs collapse safely, preventing dangerous compression of air spaces. Oxygen is stored not primarily in the lungs, but within blood and muscle tissues, where specialized proteins hold reserves far beyond those available to terrestrial mammals. Circulation changes as the dive progresses, directing resources toward essential organs while temporarily reducing blood flow elsewhere. Every aspect of the body becomes optimized for endurance beneath immense weight. Even among marine mammals, however, sperm whales do not represent the limit. That distinction belongs to beaked whales, elusive animals whose lives remain among the least understood in the ocean. Around island wall systems in the Bahamas, researchers have documented dives exceeding 2,992 meters. One recorded descent lasted 2 hours and 17 minutes on a single breath, establishing the deepest confirmed dive ever achieved by a mammal. At such depths, pressure surpasses 300 atmospheres. The surrounding water exerts a force so immense that conventional human experience offers almost no comparison. Yet the whales continue hunting within this environment, navigating through complete darkness with echolocation. Bursts of sound travel outward, strike distant objects, and return as echoes that reveal prey hidden in the blackness. Where vision becomes impossible, sound becomes perception itself. The deeper scientists have examined these dives, the more striking the pattern has become. The same factor that makes the deep ocean inaccessible to most life has also driven the evolution of some of the most sophisticated physiological systems on Earth. Pressure acts as both obstacle and opportunity. It excludes many species while rewarding those capable of adapting to its demands. Island walls provide a rare stage upon which these adaptations become visible. The dramatic depth changes concentrated beside oceanic islands allow researchers to observe animals moving between radically different pressure environments within a relatively confined geographic area. Along these submerged slopes, the boundary between worlds becomes compressed, and the biological achievements required to cross that boundary emerge with unusual clarity. The descending wall remains motionless, while above it and beside it, some of the most extraordinary divers ever produced by evolution continue their silent journeys through the weight of the sea. Long before humans possessed instruments capable of exploring the sea, the ocean was already filled with sound. Every movement, every feeding event, every call, strike, scrape, and pulse sent vibrations outward through the water. Beneath the surface, life developed within an environment where hearing often travels farther than sight. Darkness can limit vision. Distance can blur form. But sound continues onward, carrying information through the ocean with remarkable efficiency. Water transmits sound far more effectively than air. Vibrations move through it at roughly 1,500 meters per second, more than 4 times faster than they travel through the atmosphere above. Across open water, these sounds disperse through immense volumes of space. Around island walls, however, the story changes. The steep geological face alters the movement of sound just as surely as it alters the movement of water. Every ridge, ledge, overhang, and descending contour becomes part of an acoustic landscape. Sounds striking the wall are reflected back into the surrounding water. Some frequencies scatter, others concentrate. Certain areas become zones where sound is amplified, while neighboring regions fall into relative acoustic shadow. The result is a habitat layered not only by depth and current, but also by invisible patterns of resonance. Many animals appear to exploit these conditions. Communication signals that might dissipate quickly in open water can persist longer when reflected along the wall. The structure effectively extends the reach of certain sounds, creating pathways through which information travels. To the creatures inhabiting these environments, the wall is not merely a physical feature. It is part of the medium through which they perceive one another. Among the most powerful contributors to this underwater soundscape are sperm whales. Their echolocation clicks rank among the loudest biological sounds ever recorded. Individual clicks can exceed 230 decibels, powerful enough to travel vast distances through the sea. When these sounds are projected toward an island wall, echoes return from countless surfaces. Ledges, crevices, protrusions, and vertical faces each reflect sound differently. Through these returning signals, the whale constructs a detailed acoustic image of the surrounding environment, navigating through darkness using information carried entirely by vibration. Closer to the wall, a very different source of sound dominates. Colonies of snapping shrimp produce a near-continuous crackling noise that fills the water around them. Each individual snap originates from a specialized claw that closes with such speed that it creates a tiny cavitation bubble. When the bubble collapses, it generates a sharp acoustic pulse capable of stunning small prey. A single shrimp contributes only a brief sound, yet thousands acting together create a constant blanket of noise. In some locations, the collective crackling becomes one of the dominant sounds in the habitat. Deeper still, another layer emerges. Certain fishes produce low-frequency calls that lie below the limits of human hearing. These infrasonic signals travel efficiently through seawater and can propagate considerable distances. Along island walls, recordings from hydrophones have captured such sounds rising upward through the water column, carried and reflected by the geological structure itself. The complexity of these acoustic environments has occasionally led to confusion even among researchers. During the Cold War, portions of the United States Navy's underwater listening network detected unusual sounds originating from island wall regions. Some recordings were initially categorized as possible mechanical sources before later investigation revealed biological origins. Living organisms had produced signals unfamiliar enough to resemble technology. Large whales add yet another dimension. Humpback songs recorded near island walls in places such as Hawaii and Tonga interact with the surrounding topography in subtle ways. Reflections from the submerged slopes create interference patterns that may help extend the reach of the songs across greater distances. The wall becomes part of the transmission itself. Taken together, these layers form one of the richest sound environments in the ocean. Communication, navigation, hunting, and orientation all converge within a landscape shaped not only by stone and water, but by vibration. The island wall gathers sounds from many depths, redirects them through its contours, and quietly transforms the surrounding sea into a living network of echoes. Certain animals seem almost inseparable from the idea of the ocean itself. Long before modern marine biology existed, sharks were already moving through these waters, occupying ecological roles that would persist across immense stretches of geological time. Yet, even after decades of study, island walls continue to reveal behaviors in sharks that challenge assumptions about where they travel, how they interact, and what environmental forces shape their lives. Around many oceanic islands, sharks do not appear merely as occasional visitors. They gather. In some locations, the concentration is remarkable enough to alter the character of the entire ecosystem. Surveys conducted along Pacific Island walls have documented aggregations of Galapagos sharks at densities rarely, if ever, observed in open water. Dozens may occupy the same section of habitat simultaneously, their movements weaving through the currents that sweep across the submerged slopes. At first glance, the explanation appears straightforward. Island walls concentrate food, and predators gather where prey becomes abundant. Yet continued observation suggests a more complex relationship. The wall influences not only feeding opportunities, but also behavior itself. The physical structure changes how sharks interact with one another and with the environment around them. Gray reef sharks provide one of the clearest examples. Along certain Pacific Island walls, researchers have documented a distinctive display associated with territorial or defensive encounters. The shark arches its body, lowers its pectoral fins, and swims with an exaggerated rolling motion. This behavior is well known at wall sites, yet comparable populations inhabiting open water environments rarely exhibit the same display. The difference appears connected to geometry. In open water, a shark can often resolve tension simply by increasing distance. Along a steep wall, available space becomes constrained. The landscape encourages interaction in ways that open ocean habitats do not. Movement across far greater distances reveals another influence of island walls. Silky sharks undertake migrations spanning thousands of kilometers through seemingly featureless ocean. Despite these journeys, tagged individuals have demonstrated a remarkable tendency to return to specific wall sections with extraordinary precision. Year after year, the same underwater landmarks appear to draw them back. Whether guided by magnetic cues, current patterns, chemical signatures, or a combination of factors, the consistency suggests a level of spatial memory that remains only partially understood. The ecological significance of these return points becomes even clearer when considering species that have declined dramatically. Oceanic white-tip sharks were once among the most abundant large predators in tropical oceans. Today, their populations have fallen across much of their range. Island walls that formerly hosted regular aggregations now support fewer individuals, and researchers have documented corresponding shifts in local ecological dynamics. The absence of a dominant predator alters feeding relationships throughout the community, leaving subtle but measurable changes in the structure of life along the wall. Tiger sharks reveal another dimension of this influence. Tracking studies have shown individuals repeatedly moving between shallow and deeper habitats along island walls. During daylight hours, they may descend to depths approaching 300 meters before returning toward the upper water column after sunset. Similar vertical routines have not been documented with the same consistency in open ocean populations. The wall appears to provide a framework around which these daily movements are organized. Perhaps nowhere is the connection between sharks and island walls more visually striking than at locations such as Cocos Island and the Galapagos. There, large schools of hammerhead sharks gather in numbers that have become iconic within marine science. Hundreds of animals may occupy the same region of water near the wall face, maintaining formations whose purpose remains uncertain. Hypotheses involving social behavior, thermoregulation, navigation, and parasite removal have all been proposed, yet no single explanation fully accounts for the phenomenon. Taken together, these observations reveal something important. Island walls do not simply attract sharks. They shape their behavior, influence their movements, alter their interactions, and in some cases appear to drive patterns found nowhere else in the ocean. Through the presence of the wall itself, ancient predators encounter conditions that encourage new expressions of familiar instincts, creating ecological relationships as dynamic and complex as the landscape they inhabit. A cliff suggests a boundary. From the outside, an island wall appears to be exactly that, a descending face of stone separating one depth from another. Yet the rock conceals a second landscape hidden within itself. Beneath many islands, beyond the visible contours of the wall, there exists an intricate internal world of passages, shafts, chambers, and flooded corridors extending deep into the geological body of the island. These spaces originated through several processes acting across immense spans of time. Volcanic eruptions left fractures and conduits within the rock. Rainwater filtering downward dissolved soluble limestone layers. Groundwater slowly widened cracks into tunnels. Ocean circulation enlarged existing openings. Over thousands, and sometimes millions of years, isolated cavities became connected networks, transforming the interior of the island into a labyrinth beneath the sea. Among the most striking entrances to this hidden world are blue holes. From above, they often appear as dark, circular openings in otherwise shallow water, their depths difficult to judge from the surface. What begins as a vertical shaft may continue into horizontal passages that stretch far beneath the surrounding landscape. In some systems, these tunnels extend for extraordinary distances. The Andros Blue Hole Network in the Bahamas connects more than 200 kilometers of submerged cave passages, making it the longest known underwater cave system on Earth. Exploring such environments presents challenges unlike those encountered on the open wall. In open water, a diver can ascend directly toward the surface. Inside a cave, that route may disappear entirely. Passages narrow. Visibility can vanish instantly if sediment is disturbed. Complete darkness exists beyond the reach of artificial light. Every direction appears identical. For this reason, many researchers consider submerged cave systems among the most demanding environments ever entered by humans. The conditions inside differ profoundly from those outside. Ocean currents that shape the surrounding wall often fail to penetrate far into the cave network. Water can remain remarkably still. Layers of different chemistry accumulate over time, forming distinct boundaries within the flooded passages. One layer may contain relatively fresh groundwater, another may consist of dense seawater. Between them, chemical gradients create invisible transitions that can remain stable for long periods. In the deepest chambers, oxygen may disappear altogether. These anoxic zones support forms of life that operate according to very different rules from those governing surface ecosystems. Here, Certain bacteria obtain energy not from sunlight or oxygen, but from chemical reactions involving sulfur compounds dissolved in the water. Entire chemosynthetic communities can exist in these isolated regions, sustained by chemistry alone. Their existence demonstrates that life does not always require the pathways familiar from the sunlit world above. Isolation has also encouraged unique evolutionary histories. Surveys of Bahamian cave systems have revealed crustaceans, worms, and microbial communities found nowhere else on Earth. Separated from surrounding oceans for immense periods of time, these organisms followed their own evolutionary trajectories. Some retain characteristics that appear ancient when compared with their relatives in open marine habitats, offering rare glimpses into lineages shaped by long isolation. The cave walls themselves preserve records extending far beyond biology. Fossil coral remains cling to surfaces now submerged in darkness. Mineral deposits mark former water levels. Geological layers document repeated rises and falls of the sea across hundreds of thousands of years. Each chamber functions as a natural archive, storing evidence of environmental change long after those events disappeared from the surface world. During the last ice age, global sea levels stood approximately 120 meters lower than they do today. Many passages now flooded with seawater were then dry caves filled with air. Stalactites formed from dripping groundwater. Animals entered these chambers. In some locations, human remains and artifacts have been discovered within cave systems that later flooded as glaciers melted and the ocean gradually rose. The still waters that eventually filled these spaces often preserved what entered them. In oxygen-poor conditions, decomposition slowed dramatically. Bones, sediments, and traces of ancient landscapes remained protected within the darkness. Hidden inside the island itself, beyond the visible wall, and beyond the reach of ordinary currents, these submerged caverns continue to hold a layered record of geology, climate, evolution, and time. Every living community leaves traces of itself behind. A forest alters the soil beneath it. A river carries dissolved minerals from the mountains through which it flows. In the ocean, life and geology also leave signatures, though they are often invisible. Around island walls, these signatures accumulate into something remarkably distinctive, a chemical landscape that extends far beyond the stone itself and quietly influences everything that moves through the surrounding sea. Water arriving from deeper layers carries more than temperature and nutrients. Suspended within it are nitrates, phosphates, and silicates, compounds that have spent long periods circulating through the depths. When ocean circulation lifts this water toward shallower levels around island walls, those compounds enter environments where sunlight remains abundant. The result is a sudden availability of resources that microscopic organisms can rapidly exploit. Phytoplankton respond first. These drifting photosynthetic organisms are tiny, yet collectively they form the foundation of nearly every marine food web. When nutrients become available, their populations expand. Blooms spread through the water above and around the island. From orbit, satellites measuring chlorophyll concentrations repeatedly detect these productive regions as subtle halos surrounding oceanic islands. Against the relatively nutrient-poor waters of the open sea, the chemical influence of the wall becomes visible on a planetary scale. The consequences ripple outward through the ecosystem. Zooplankton gather where phytoplankton flourish. Small fishes follow the concentrations of plankton. Larger predators arrive behind them. What appears at first to be a simple chemical enrichment becomes the beginning of a chain reaction extending through every level of the food web. The chemistry generated by the wall does not originate solely from nutrients rising from below. The living community attached to the wall contributes its own chemical imprint. Organisms feed, grow, reproduce, and eventually die. Through these processes, they release dissolved organic carbon into the surrounding water. This material forms a subtle gradient that accumulates along the vertical face, creating a chemical pathway extending outward into the sea. For many marine animals, smell is not a secondary sense. It is a primary means of navigation. Sharks, in particular, possess an extraordinary sensitivity to dissolved compounds. Researchers studying their movements have found evidence suggesting that chemical plumes originating around island walls function almost like navigational markers. Long before a shark can detect the island visually, it may already be following gradients in water chemistry that reveal the wall's presence. Corals and other attached organisms contribute additional layers to this chemical complexity. Many deep-water species manufacture compounds known as secondary metabolites. These molecules are not directly involved in basic metabolism but serve defensive, competitive, or ecological functions. Over the past several decades, scientists have discovered that some possess properties of considerable medical interest. Certain compounds isolated from deep water corals and related organisms have demonstrated anti-inflammatory activity, while others have shown potential in cancer research and pharmaceutical development. Part of what makes these discoveries so valuable is their uniqueness. Species living along island walls often occupy environmental conditions unlike those found elsewhere. At depths between 200 and 400 meters, communities experience combinations of pressure, nutrient availability, and biological interaction that are difficult to replicate. The chemistry produced by such organisms reflects those specialized conditions, resulting in molecules rarely encountered in other habitats. The geology of the wall contributes as well. Volcanic rock slowly releases trace elements into the surrounding water. Iron, manganese, zinc, and other micronutrients enter circulation in small quantities. Although present at low concentrations, these elements can have outsized biological effects. In regions where such nutrients are scarce, even slight additions can enhance productivity and influence ecological dynamics. Together, these biological and geological processes create a chemical signature that extends far beyond the island itself. Currents carry dissolved compounds outward for hundreds of kilometers. The wall leaves an invisible imprint upon the surrounding ocean, a drifting chemical shadow moving through the water long after the stone has disappeared beyond the horizon. Creatures encountering this signal may never see the island, yet they are already responding to its presence through the language of chemistry. Across enough time, isolation becomes a creative force. A population begins as part of something larger, sharing genes with relatives spread across a broader landscape. Then a barrier appears. The exchange slows. Small differences accumulate. Generation after generation, those differences deepen until a new biological path emerges. Evolution has repeated this process countless times throughout Earth's history, and island walls provide some of the most remarkable settings in which it continues to unfold. At first glance, the surrounding ocean appears to offer unlimited connection. Water stretches uninterrupted in every direction, seemingly allowing organisms to move freely between islands. Yet the reality is more complicated. For many species, the open sea functions less like a highway and more like a vast expanse of unsuitable habitat. The wall may provide everything an organism requires, while the surrounding waters provide very little. In such circumstances, distance is measured not only in kilometers, but in ecological compatibility. This combination of isolation, stability, and environmental complexity has allowed island walls to become engines of endemism. Species living on one wall often share ancestry with populations elsewhere, yet over long periods they diverge. Currents differ, depth profiles vary, geological structures create unique conditions. The result is a gradual accumulation of biological distinctions that become visible in anatomy, behavior, genetics, and ecological role. Researchers studying the Pacific have documented striking examples of this process. Johnston Atoll hosts Gorgonian coral species found nowhere else on Earth. Although surrounded by ocean theoretically connected to countless other habitats, these corals remain genetically isolated. Their lineage has followed its own evolutionary trajectory, shaped by local conditions and separated from close relatives by barriers invisible to the eye. Fish communities reveal the same pattern on a broader scale. The Hawaiian Islands possess one of the highest rates of marine endemism ever recorded. More than a quarter of their fish species occur nowhere else in the world. Many evolved in association with the island walls that define the region's underwater geography. Over immense spans of time, these populations adapted to local conditions until they became distinct expressions of life found only within that isolated archipelago. The process does not stop at the level of species. Genetic studies increasingly reveal divergence among populations that outwardly appear identical. Two groups of the same fish may inhabit neighboring islands, separated by relatively modest distances, yet analysis of their DNA shows long periods of limited exchange. Despite the apparent continuity of the surrounding ocean, biological separation persists. Part of the explanation lies in the specialized nature of wall habitats. Organisms adapted to a particular combination of current strength, nutrient availability, depth range, and ecological interactions may find it difficult to establish themselves elsewhere. Even when larvae drift between islands, survival is not guaranteed. The receiving environment may differ enough to prevent successful colonization. Meanwhile, the wall continually generates selective pressures rarely encountered in other marine settings. Species must navigate interactions with organisms arriving from different depth zones. Predators, prey, and competitors meet in unusual combinations. Environmental gradients change rapidly over short distances. Every generation experiences a complex mosaic of challenges that encourages adaptation. These conditions help explain why scientific surveys continue producing discoveries at a remarkable pace. For more than 3 decades, researchers exploring island wall habitats have described new species year after year. The trend has shown little indication of slowing. Each expedition reveals organisms that had escaped previous observation, despite advances in diving technology, remotely operated vehicles, and genetic analysis. What emerges from this pattern is a portrait of evolution as an ongoing process rather than a completed story. Island walls are not simply repositories of rare species. They are places where biological diversity continues to be generated. Along these submerged slopes, isolation and connection exist simultaneously, creating conditions that allow life to experiment, diverge, and persist in forms found nowhere else. Even now, hidden among the countless niches scattered across the world's island walls, evolutionary journeys continue in quiet obscurity, unfolding one generation at a time. A beam of sunlight entering the ocean begins to change almost immediately. The transformation is gradual, yet relentless. Colors fade one by one, brightness diminishes, and the familiar visual world of the surface gives way to something increasingly selective. Along an island wall, This transformation unfolds with unusual intensity because an immense range of lighting conditions is compressed into a relatively short vertical distance. Near the surface, sunlight arrives in its fullest form. Reds, oranges, yellows, greens, and blues illuminate the water column together, yet water is not equally transparent to every wavelength. Red light disappears first, absorbed within the upper layers. As depth increases, orange follows, then yellow. The descent continues through a narrowing spectrum until blue becomes the dominant remaining color. By the time depths approach 200 meters, almost no sunlight remains at all. The transition from brightness to near darkness has occurred within the span of a single wall. For the organisms living there, vision becomes a highly specialized adaptation. Species occupying different depths do not simply see the same environment under dimmer conditions. They often perceive fundamentally different worlds. A fish inhabiting depths around 50 meters may possess eyes optimized almost entirely for blue wavelengths. Colors visible to surface species no longer play a meaningful role in its visual experience. Its sensory system has evolved around the specific light environment available at that depth and nowhere else. This continuous gradient creates a mosaic of visual habitats stacked upon one another. Every zone favors different solutions to the challenges of survival. Eyes change shape, sensitivity, and structure according to the available light. The wall becomes a living gallery of evolutionary experiments in perception. Particularly fascinating is the mesophotic region, sometimes called the twilight zone of the wall. Extending roughly between 30 and 150 meters, this environment receives only a fraction of the sunlight present near the surface. Here, certain fish species possess visual systems of remarkable sophistication. Some contain 4 distinct types of color receptors rather than the 3 commonly found in human eyes. In these dim blue conditions, their ability to distinguish subtle variations in color may exceed our own, allowing them to navigate a world of visual information largely invisible to surface observers. As the wall descends further, another strategy emerges. In the deep sections, many fishes develop disproportionately large eyes. The available light becomes so scarce that maximizing photon collection becomes essential. Some species devote an astonishing fraction of their head structure to vision, with eyes occupying roughly a quarter of total head volume. Every additional bit of light becomes valuable. Evolution rewards any adaptation capable of extracting information from the darkness. This environment drives a continual visual competition. Predators evolve greater sensitivity to faint movement and subtle contrast. Prey evolve methods of concealment adapted to those same sensory systems. In many cases, the contest extends beyond ordinary reflection of ambient light and enters the realm of bioluminescence. Numerous wall-dwelling species produce their own light through specialized organs. These signals serve many purposes, including communication, recognition, attraction, and camouflage. One particularly elegant strategy is known as counter-illumination. Rather than hiding in darkness, an organism generates light along its underside that matches the faint illumination descending from above. To a predator looking upward, the animal's silhouette disappears into the surrounding glow, blending almost perfectly with the background. What makes the island wall extraordinary is the precision with which these adaptations are distributed. A relatively small change in depth alters the quality, intensity, and spectral composition of available light. The visual environment at one level differs subtly from the visual environment only a short distance away. Over evolutionary time, organisms have responded to these differences with extraordinary specificity. The result is a descending sequence of worlds, defined not by geography, but by perception. Along the wall, life has adapted to every stage of the fading light, creating communities whose eyes, behaviors, and signals are calibrated to the exact visual conditions of their particular place within the spectrum of the sea. Not every boundary in the ocean can be seen. Some exist as invisible layers suspended in the water itself, dividing one world from another with remarkable precision. Along island walls, few of these boundaries are more influential than the thermocline, a narrow zone where temperature changes so rapidly that it reshapes the lives of nearly everything around it. The ocean generally becomes colder with depth, but this transition is often gradual across vast stretches of open water. Island walls compress the process. Within a relatively short descent, warm surface layers can give way to dramatically colder water. In many wall environments, the thermocline appears between roughly 20 and 50 meters. Across a vertical distance of only a few meters, temperatures may fall by as much as 10 degrees Celsius, creating a sharp thermal divide suspended between the shallows and the depths. For those moving through the water, the transition is unmistakable. A descending diver encounters a sudden change that feels almost like passing through an unseen curtain. The water becomes colder, but the sensation extends beyond temperature alone. Visibility often shifts. Light behaves differently. Refraction changes subtly as water densities vary. Even buoyancy feels altered, as denser cold water provides a slightly different physical support. The ocean remains continuous, yet the experience of inhabiting it changes abruptly. Marine life responds to this boundary with equal sensitivity. Many reef fishes spend their entire lives above the thermocline. Their physiology evolved within stable, warm conditions, and crossing into colder water imposes costs that can affect metabolism, immune function, and reproduction. Although the deeper habitat may be physically nearby, it remains biologically inaccessible. The thermocline functions as an invisible ceiling, quietly restricting movement without the need for any physical barrier. The same boundary acts in reverse for organisms adapted to cooler depths. Species comfortable below the thermocline often remain constrained there, their bodies optimized for conditions that differ dramatically from those near the surface. Between these communities lies a narrow thermal frontier separating ecosystems that may be only a short swimming distance apart, yet experience profoundly different realities. Some predators have learned to exploit this arrangement. Rather than committing fully to either side, they position themselves directly along the boundary. Hovering within the temperature gradient, they gain access to prey arriving from both directions. Organisms descending from warmer water encounter unfamiliar conditions. Others ascending from below enter a zone of transition. The thermocline becomes not merely a divider, but an ecological meeting place where opportunities concentrate. Yet the boundary is not fixed. Seasonal changes alter its position. During cooler periods, surface waters lose heat and the thermocline can descend deeper along the wall. Species that normally remain separated may briefly overlap. Habitats expand or contract. Subtle shifts in temperature redistribute entire communities, changing feeding relationships and movement patterns throughout the ecosystem. Even more dramatic are cold water intrusion events. At certain times, unusually strong upwelling can force large volumes of deep water upward along the wall. The cold layer rises toward the surface, carrying conditions normally associated with far greater depths. Organisms adapted to warmth may suddenly find themselves immersed in temperatures beyond their tolerance. These events reveal an often overlooked reality of marine ecology. Environmental stress is not always driven by excess heat. Along island walls, cold can be equally disruptive. Shallow corals that flourish under stable conditions may experience significant mortality when deep water surges upward in sufficient volume. Communities accustomed to one thermal regime can be replaced temporarily by species better suited to the altered conditions. Through all of this, the thermocline remains an architect of invisible geography. It shapes where organisms live, where they hunt, where they reproduce, and where they cannot go. Along the face of an island wall, temperature becomes more than a physical property of water. It becomes a structure in its own right, a shifting framework that quietly organizes life from the sunlit surface to the colder realms below. Far below the depths where most divers turn back, the island wall continues descending into water that remains largely beyond direct human observation. The reef communities above gradually fade from view, replaced by a predator assemblage that is older, larger, and in many respects far less understood. These hunters occupy a realm where light is scarce, pressure is immense, and survival depends upon adaptations refined across vast stretches of evolutionary time. Among the most remarkable residents of this deeper zone is the six-gill shark. Its lineage reaches back roughly 200 million years, preserving a body plan that emerged long before many modern marine ecosystems existed. During daylight hours, six gills often occupy depths between 200 and 500 meters along island walls. As darkness spreads through the upper ocean, they begin a slow ascent. Individuals have been documented rising to depths of roughly 30 meters before returning downward again before sunrise. These movements are not occasional excursions. Tracking studies reveal a daily rhythm. Along island walls in British Columbia, blunt-nose, six-gill sharks, equipped with depth recording tags, have demonstrated vertical migrations exceeding 400 meters within a single cycle. The wall functions as a hunting corridor extending through multiple ecological zones. Rather than moving across great horizontal distances, these predators travel up and down the submerged landscape, following opportunities distributed through the water column. The dimensions of these animals reinforce their status within the ecosystem. Mature individuals can exceed 5 meters in length and weigh more than 600 kilograms. Yet despite their size, they often remain hidden from human awareness, spending much of their lives beyond the depths regularly visited by divers or conventional surveys. Even deeper, another predator patrols the colder reaches of northern island walls. The Pacific sleeper shark is among the largest predatory fishes inhabiting these environments. Reaching lengths of approximately 7 meters, it moves through depths exceeding 2,000 meters with a quiet persistence that remains difficult to study. Researchers know surprisingly little about its behavior. Encounters are rare, observations limited, and much of its life unfolds beyond the reach of routine exploration. One of the few direct recordings captured a sleeper shark feeding on the remains of a sperm whale that had settled against an island wall, revealing its role as both hunter and scavenger within the deep ecosystem. Other ancient lineages contribute additional mysteries. Broadnose seven-gill sharks, close relatives of some of the earliest shark groups, display behaviors that remain unexplained. In certain island wall cave systems, researchers have documented gatherings of dozens of individuals resting together during daylight hours. Groups approaching 50 sharks have been observed occupying the same sheltered spaces. The purpose of these aggregations remains uncertain. Current scientific literature offers no definitive explanation for why these predators repeatedly assemble in such numbers. Sharks are not the only dominant hunters inhabiting the deeper sections of island walls. Large predatory groupers also reach extraordinary proportions in these environments. On Indo-Pacific wall systems, deep-dwelling species often grow far larger than their counterparts, inhabiting shallower reefs. The Queensland grouper, documented along outer wall habitats associated with the Great Barrier Reef region, can exceed 2.5 meters in length and approach 400 kilograms in mass. Such dimensions place it among the largest bony fishes associated with coral reef ecosystems. What makes these observations especially significant is how little of this environment has been systematically surveyed. Below approximately 100 meters, comprehensive biological exploration remains remarkably limited. The technology required for routine investigation at such depths has existed for only a relatively short period. Remotely operated vehicles, advanced submersibles, and deep camera systems have only recently begun to reveal the communities occupying these darker sections of the wall. Each new survey tends to produce unexpected results. Species appear outside their previously known ranges. Behaviors emerge that were never recorded in shallower environments. Established assumptions require revision. The deeper reaches of island walls remain among the least familiar predator habitats on Earth, a place where ancient hunters continue their movements through the darkness, largely unseen, while science slowly expands its understanding of the lives unfolding far below. Most archives are built intentionally. Books are placed on shelves, records are stored in vaults, and documents are organized so future generations can retrieve them. Island walls created a different kind of archive. No intelligence designed it, yet over immense spans of time the wall accumulated layer upon layer of information, preserving traces of climates, oceans, ecosystems, and landscapes that vanished long before humans arrived to study them. A descending wall face is not merely a geological structure, it is a timeline. Every ledge, terrace, mineral layer, and fossil deposit represents a specific chapter in Earth's history. Unlike many environments where erosion or biological activity erase older records, island walls often preserve them with remarkable clarity. The result is a vertical archive stretching through hundreds of thousands of years. One of the most visible entries in this archive appears as fossil coral terraces etched across the wall. These horizontal bands mark former sea levels, preserving the position of ancient coastlines with surprising precision. When sea level remained stable for long periods, coral reefs flourished near the surface. As conditions changed and oceans rose or fell, those reefs were abandoned and preserved. What remains today are submerged terraces that record where the sea once stood. Along Caribbean island walls, one particularly significant terrace occurs at roughly 120 meters below the modern surface. This feature corresponds to the last glacial maximum, a period approximately 20,000 years ago when enormous ice sheets locked vast quantities of water on land. Global sea levels fell dramatically. At that time, locations that now lie deep underwater occupied very different positions within the landscape. The implications are striking. The seafloor currently resting at 120 meters depth was once part of a shoreline environment. Waves broke there. Sediments accumulated there. The wall extending below that level stood exposed to air rather than submerged beneath seawater. Entire portions of the modern underwater landscape existed as dry land during that colder chapter of planetary history. Evidence for these transformations extends beyond the wall face itself and into the cave systems hidden within the island. Deep inside flooded passages, researchers have discovered stalactites hanging from cave ceilings. These formations develop only when water drips through air-filled chambers over long periods. Their presence inside submerged caves provides unmistakable proof that those spaces once existed above sea level. When oceans rose following the end of the last ice age, seawater gradually flooded these chambers. The stalactites remained behind as geological witnesses, preserving a timeline of environmental change. Through dating techniques, scientists can determine when the caves were dry and when rising seas finally reclaimed them. Sediments collected from island wall environments extend the record even further. Drill cores, extracted from accumulated deposits, contain pollen grains, microscopic fossils, and chemical markers spanning roughly 500,000 years. Layer by layer, these sediments preserve evidence of repeated glacial and interglacial cycles. They document shifts in vegetation, ocean circulation, biological productivity, and climate conditions across periods far older than recorded human history. Ancient coral skeletons contribute another remarkable source of information. As corals grow, they incorporate chemical signatures from the surrounding seawater into their calcium carbonate structures. Long after the living organism has died, those signatures remain preserved. Scientists can analyze them to reconstruct past ocean temperatures and chemistry. In effect, the coral becomes a natural thermometer, extending deep into the geological past. The archive also records biological change. Fossil layers reveal moments when one ecological community disappeared and another replaced it. These transitions often align with broader environmental events, including shifts in ocean chemistry, climatic reorganizations, and extinction episodes documented elsewhere around the globe. Taken together, these records transform the island wall into something far greater than a habitat. It becomes a chronicle written in stone, coral, minerals, and sediment. Long before humans developed language, the wall was already preserving the story of changing oceans and changing worlds, storing each chapter patiently within its layers while the planet continued its slow passage through time. An island wall may appear, at first glance, to be a collection of individual organisms sharing the same space. Fish move through the water. Corals anchor themselves to stone, sponges filter passing currents, and crustaceans occupy cracks and ledges. Yet the true structure of the wall is not built from individuals alone. It emerges from relationships, thousands of biological partnerships woven together into networks so intricate that the survival of one species often depends upon the continued existence of another. Among the most visible examples are the cleaning stations scattered across the wall face. These are not random locations. They occupy specific rocks, ledges, and crevices that remain active year after year. Large fishes approach them deliberately, often slowing their movements and adopting recognizable postures. Waiting, there are cleaner fish and cleaner shrimp, small organisms that remove parasites, damaged tissue, and unwanted hitchhikers from their larger visitors. What makes these sites remarkable is the degree of cooperation involved. Predators capable of consuming the cleaners often refrain from doing so. The relationship provides a service valuable enough to override immediate feeding opportunities. Over time, an entire network of fixed service points develops across the wall. Hundreds of species may visit these locations, creating a biological infrastructure that resembles an economy more than a simple feeding interaction. As the wall descends beyond the reach of sunlight, similar relationships continue in darkness. Deep water, cleaner shrimp, operate where visual signals become unreliable. Instead of depending upon color or body posture, they rely on chemical cues and physical contact. Fish approaching these stations announce their presence through touch and scent. The exchange proceeds, much as it does in shallower water, demonstrating that cooperation can persist even where vision has largely disappeared from the interaction. Elsewhere on the wall, giant barrel sponges perform another essential function. Some individuals grow to more than 2 meters in diameter and may survive for over 2,000 years. Their bodies continuously filter extraordinary volumes of seawater. A single sponge can process more than 20,000 liters each day, extracting microscopic particles and redistributing nutrients throughout the surrounding community. The influence of these ancient organisms extends far beyond filtration. Their internal chambers provide shelter for specialized residents. Certain goby species spend their entire lives inside barrel sponges, rarely venturing beyond the protective structure. The relationship is so complete that the fish cannot survive without their host. In return, waste produced by the gobies contributes nutrients that support microbial communities within the sponge, creating benefits that flow in both directions. The wall contains countless examples of such dependencies. Some are obvious, while others remain hidden within microscopic interactions. Together, they create pathways through which energy and resources circulate, linking organisms that may appear unrelated when viewed separately. One of the most significant transitions occurs near the lower edge of the illuminated zone. In shallower waters, many corals maintain a partnership with microscopic algae living within their tissues. These algae perform photosynthesis and provide energy to their hosts. As depth increases and sunlight diminishes, that arrangement becomes increasingly difficult to sustain. Eventually, a threshold is crossed. Below this level, corals abandon dependence on photosynthetic partners and adopt a different strategy. They become active particle feeders, capturing organic material carried by currents rather than relying upon sunlight-driven productivity. This transformation creates the mesophotic reef, a community occupying depths where conditions differ profoundly from those above. Within this transition zone, coral species appear that are rarely encountered elsewhere. Their existence reflects the unique balance of light, nutrients, and water movement created by the wall itself. They are products not simply of physical conditions, but of relationships, adaptations, and exchanges that have unfolded across immense spans of evolutionary time. Seen from this perspective, the wall functions less like a collection of species and more like a living network. Every cleaner station, sponge chamber, coral colony, and hidden partnership contributes to a larger system whose productivity emerges from cooperation as much as competition. Life along the wall persists because countless organisms have learned, in different ways and at different depths, how to survive together. Long before charts, compasses, or written records described these waters, island walls were already woven into human experience. Their presence shaped travel routes, fishing grounds, ceremonies, and survival itself. The sea surrounding an oceanic island may appear vast and uniform from above, yet generations of coastal communities learned that the wall marked a boundary where life gathered, currents shifted, and opportunities emerged. Across centuries, knowledge accumulated not in libraries, but in memory, observation, and practice, passed from one generation to the next through stories carried across the water. Today, traces of those relationships remain preserved within the geology of the walls themselves, hidden in places where stone, sediment, and seawater have quietly protected evidence of human lives. Among the most remarkable archives are the flooded cave systems associated with Bahamian blue holes. Within these submerged chambers, archaeologists have recovered human skeletal remains dating to roughly 1,000 years before European contact. These individuals entered landscapes that looked very different from those visible today. Sea levels stood elsewhere, passages remained accessible, and portions of cave systems that now require specialized diving equipment were once connected to dry or partially dry environments. The caves became natural vaults, preserving evidence that would have disappeared almost anywhere else. The Lucayan people, the indigenous inhabitants of the Bahamas, developed relationships with these spaces that extended beyond practical use. Artifacts recovered from blue hole environments suggest ritual activity within cave systems that later vanished beneath rising seas. In the oxygen-poor sediments resting at the bottom of these chambers, objects remain protected from the decay that would normally erase them. Pottery fragments, tools, and cultural materials survived in extraordinary condition, allowing glimpses into traditions that endured for generations before outside contact transformed the region. Similar stories unfold across the wider Pacific, where island wall environments supported societies separated by immense distances of open ocean. The productivity concentrated around these walls sustained fishing economies capable of supporting complex communities across thousands of kilometers. Long before modern navigation, Pacific voyagers crossed enormous stretches of water and returned with remarkable consistency. Their success depended not only on stars, winds, and waves, but also on intimate familiarity with the biological rhythms associated with island walls. Seasonal migrations, shifting currents, and changes in species abundance became part of an accumulated body of ecological knowledge refined across countless observations. Modern researchers increasingly recognize that this traditional knowledge represents a sophisticated data set gathered over immense spans of time. Observations concerning migration timing, current behavior, and species distribution often align closely with findings produced by contemporary marine science. What survived through oral traditions sometimes describes patterns that formal scientific surveys documented only recently. The wall, therefore, preserves not merely biological history but intellectual history as well, evidence of sustained human attention directed toward one of the ocean's most complex environments. Beneath these same waters lies another archive. The currents that concentrate life around island walls also create navigational danger. Throughout history, vessels approaching unfamiliar islands encountered powerful flows, sudden weather changes, and coastlines that offered little margin for error. As a result, the bases of many island walls have become repositories of shipwrecks from different eras. Trading vessels, warships, merchant ships, and fishing boats rest in darkness where the wall descends into deeper water. Cold conditions and limited disturbance often preserve them for centuries, turning the seafloor into a layered record of human movement across the oceans. Archaeological surveys have even identified ancient stone anchors associated with voyages that occurred long before surviving historical accounts describe them. Resting quietly against the submerged architecture of the islands, these objects suggest journeys, exchanges, and maritime connections whose full stories remain unknown, preserved within the same walls that have watched both natural and human history unfold side by side across the ages. Island walls reveal their importance most clearly when parts of them begin to fail. In healthy conditions, the countless organisms attached to the wall appear so seamlessly connected that it is easy to mistake stability for permanence. Yet the balance maintained across these vertical ecosystems depends on living structures that require time, space, and continuity. When one component is damaged, the effects rarely remain confined to a single species or depth. Instead, consequences move outward through the wall like a slow current, touching communities that may seem unrelated at first glance. Among the most destructive disturbances are outbreaks of crown of thorns, starfish. Under natural conditions, these animals are part of reef ecosystems, feeding on coral without overwhelming it. Around heavily developed islands, however, nutrient pollution entering coastal waters can alter ecological balances and increase the survival of starfish larvae. Population explosions sometimes follow. Vast numbers of starfish advance across wall faces, consuming living coral tissue faster than replacement can occur. Colonies that required generations to establish can be stripped bare within surprisingly short periods, exposing the limestone framework beneath. The newly exposed surface does not remain empty for long. Algae quickly occupies available space, spreading across the wall and forming dense coverings that transform the character of the habitat. Once established, these algal communities create conditions unfavorable for coral recovery. Young coral larvae struggle to attach themselves to occupied surfaces, and certain algae release compounds that interfere with coral development. What begins as a temporary loss of coral can therefore become a long-lasting ecological shift. The wall retains its physical shape, yet its biological identity changes profoundly. As coral disappears, the intricate architecture that sheltered countless organisms begins to simplify. Small fish lose hiding places, invertebrates lose feeding surfaces, reproductive sites vanish. Species that relied on narrow crevices and branching structures become less abundant, followed by predators that depended upon them. Scientific surveys conducted on damaged walls have documented measurable declines in biodiversity within only a few years, demonstrating how strongly the community depends upon structural complexity created by living organisms. Additional pressures arrive from the land above. Sediment released through construction, deforestation, or coastal modification drifts seaward and settles downward. Gravity carries much of this material along the wall face, where it accumulates in ledges, hollows, and deeper recesses. Filter-feeding animals struggle beneath the constant burden of fine particles. Cave entrances become partially obstructed. Delicate biological processes shaped by clear water begin to change as suspended material alters both chemistry and visibility. Disease can intensify these losses. In parts of the Caribbean, black band disease has become one of the most recognizable coral infections affecting wall communities. The disease appears as a dark, advancing boundary, moving across living colonies. Behind it remains only dead skeleton. Progress can occur at approximately 1 centimeter each day, a pace that seems slow until viewed against colonies that required centuries to attain their size. The movement is steady, patient, and relentless, consuming living tissue layer by layer. What makes these losses especially significant is the timescale required for recovery. Many of the organisms that define deeper wall environments grow with extraordinary slowness. Ancient black coral colonies and massive Gorgonian structures accumulate their size across spans of time, measured not in decades, but in centuries. When such communities are damaged, replacement cannot occur within ordinary human planning horizons. The wall remembers disturbance for generations. Evidence of this long memory can still be seen on sections of wall in the Florida Keys, damaged by ship groundings during the 1980s and 1990s. Decades later, scars remain visible. Biological communities have returned in part, yet the original complexity has not fully reassembled. The lingering marks serve as quiet reminders that while island walls possess immense resilience, their living architecture is built slowly, and once broken, the passage back toward balance unfolds with the same patient pace that created the wall community in the first place. As daylight fades from an island wall, the transformation begins long before complete darkness arrives. Colors soften, shadows lengthen, and the familiar inhabitants of the daytime community gradually withdraw from open view. What appears from a distance to be the same environment is already becoming something fundamentally different. Marine biologists often describe the day wall and the night wall as separate ecosystems occupying the same physical structure, because the cast of active species, the flow of energy, and even the dominant behaviors change so completely after sunset that the resemblance between the two can seem surprisingly small. Among the first participants in this nightly transition are parrotfish. Throughout the day, they graze openly across reef surfaces, their movements helping shape the community around them. As darkness approaches, they settle into sheltered positions and begin producing a remarkable form of protection. Specialized glands near the mouth secrete a mucus envelope that gradually surrounds the resting fish. The process requires roughly 30 minutes to complete. By morning, another 30 minutes may pass before the cocoon dissolves. The structure appears delicate, yet it serves an important purpose, masking chemical cues that nocturnal predators might otherwise use to locate a sleeping fish hidden among the contours of the wall. At nearly the same time, countless coral polyps emerge from colonies that seemed motionless during the day. Tiny feeding tentacles extend into the water, converting the wall into an immense living surface dedicated to capturing drifting particles. What appeared under sunlight as stone and structure reveals itself after dark as a dense landscape of feeding organisms. Across vast stretches of the wall, millions of microscopic predators begin harvesting nourishment from passing currents, creating a level of biological activity invisible to daytime observers. As the night deepens, organisms that remained concealed through daylight hours move into open space. Studies of island wall environments suggest that visible invertebrate biomass after dark can exceed daytime levels by a factor of 10. Crustaceans, worms, mollusks, and countless smaller creatures emerge from crevices and hidden chambers. The wall becomes crowded with movement. Life that spent the day concealed within narrow spaces now occupies surfaces, ledges, and water immediately adjacent to the rock face. Among the most striking visitors are Spanish dancer nudibranchs. Reaching lengths of approximately 60 centimeters, they rank among the largest sea slugs in the world. During daylight, they remain hidden within protected recesses. At night, they venture outward and sometimes leave the wall entirely, swimming through open water with broad, undulating motions. Their flowing movements create an appearance unlike almost any other marine animal, a behavior rarely witnessed under the sun. Predators also alter their routines. Moray eels, often regarded as solitary and territorial inhabitants of crevices and caves, participate in behaviors that reveal unexpected cooperation. At certain island wall environments, researchers documented partnerships between moray eels and roaming groupers. The relationship was formally described during the first decade of the 21st century in the Red Sea. Eels probe narrow openings inaccessible to larger fish, flushing hidden prey into open water where waiting groupers can pursue it. Both hunters benefit, demonstrating a level of coordination that reshapes assumptions about how reef predators interact. Beyond the wall itself, the surrounding water acquires an entirely different character. Disturbances created by waves, currents, and swimming animals trigger flashes from bioluminescent dinoflagellates suspended in the water column. Blue and green points of light appear and vanish in shifting patterns, tracing invisible movements through the darkness. The display drifts beside the wall like a living constellation suspended beneath the sea. Repeated night surveys have consistently revealed species, interactions, and behaviors absent from daytime records. Each expedition adds evidence that daylight observations capture only part of the story. The wall that emerges after sunset is not merely a darker version of the one seen beneath the sun. It is a fuller expression of the ecosystem itself, revealing layers of activity, cooperation, feeding, and movement that remain hidden until darkness quietly settles across the ocean. Far beyond the view of any shoreline, the influence of an island wall continues to travel through the ocean. Water moving past a remote volcanic island carries traces of the wall with it, subtle signatures of chemistry, biology, and energy that spread outward into surrounding seas. What appears at first to be an isolated geological feature is in reality part of a much larger system. Island walls function as exchange points where the deep ocean and the sunlit surface interact with unusual intensity, creating consequences that extend far beyond the islands themselves. One of the most important of these exchanges involves the movement of carbon through the ocean. Near productive wall environments, microscopic plants flourish in nutrient-rich surface waters, supporting food webs that stretch from tiny zooplankton to large predators. Yet not all of this biological material remains near the surface. Dead organisms, waste particles, and fragments of organic matter gradually sink downward through the water column. Oceanographers refer to this process as the biological pump, a mechanism through which carbon captured near the surface is transported into deeper layers of the sea. Around island walls, this transfer often operates with unusual efficiency. Productivity generated near the wall creates a steady rain of organic material descending toward the abyss below. Although these environments occupy only a tiny fraction of the ocean's total area, survey data suggests they contribute a disproportionately large share of biological carbon export relative to their size. The wall acts almost like a conveyor between worlds, linking sunlight-driven production above with deep ocean storage far below. The influence does not end when this material reaches the depths. Communities living on the abyssal floor beneath active wall systems benefit from a more reliable supply of food than comparable regions located far from island influence. On the otherwise sparse deep seabed, even modest increases in falling organic matter can transform ecological conditions. Organisms that depend upon this descending nourishment often occur in greater abundance beneath productive wall environments, creating pockets of biological richness in places that might otherwise remain comparatively barren. At the same time, island walls serve as navigational landmarks within the vastness of the open ocean. For migratory species crossing immense distances, the sea can appear remarkably uniform, yet walls generate distinct chemical, acoustic, and biological signatures that extend far beyond their physical boundaries. Sea turtles moving across ocean basins, sharks undertaking seasonal migrations, whales traveling between feeding and breeding grounds, and schools of tuna traversing open water all encounter these signals. Long before an island becomes visible, the ocean itself begins announcing its presence. These journeys create connections between distant walls separated by extraordinary distances. Migratory animals transport genes, nutrients, and ecological relationships from one region to another. A shark born near one island chain may later contribute to populations associated with another. Sea turtles linking distant nesting and feeding grounds help maintain genetic continuity across enormous expanses of water. Through these travelers, events occurring at one wall can influence biological communities thousands of kilometers away. Yet the same ocean that enables connection also creates separation. As explored in earlier passages, many species lack the ability to cross deep channels between island groups. For them, the open water functions as a barrier rather than a bridge. Populations become isolated, evolutionary paths diverge, and unique species emerge. The processes of connection and separation occur simultaneously, shaping the diversity that defines island wall ecosystems. Across the Pacific, this pattern becomes especially visible. The Hawaiian chain, the Mariana Arc, the islands of Micronesia, and countless other oceanic formations create a scattered network of biological nodes extending across the largest ocean on Earth. Each wall functions as a local center of productivity and evolution, yet all remain linked through the movements of water, carbon, sound, and life itself. Together they form a hidden framework beneath the ocean's surface, helping organize the distribution of marine life across an immense blue world that is far more connected than it first appears. There was a time when the island wall simply vanished from human knowledge. A swimmer could descend through clear water until lungs demanded a return to the surface, and beyond that limit, the wall continued downward into mystery. For thousands of years, everything beneath those reachable depths remained hidden. The wall existed, currents flowed across it, predators hunted along its face, and entire communities lived in darkness. Yet human understanding stopped within the narrow upper layer, accessible through breath alone. Even the most skilled, traditional divers could rarely remain long beyond depths of roughly 20 to 30 meters. Below that threshold, observation gave way to imagination. The wall extended downward, but no direct witness could follow it far enough to understand its true scale. For much of history, knowledge of island walls was therefore incomplete, confined to a thin, illuminated skin at the ocean's surface, while the overwhelming majority of the ecosystem remained unseen. The first major expansion came during the middle of the 20th century with the development of modern scuba equipment. For the first time, divers could remain underwater for extended periods while carrying their own supply of breathing gas. Recreational exploration reached depths around 40 meters, while highly trained technical divers eventually pushed farther, sometimes descending between 60 and 80 meters. These advances revealed astonishing complexity, yet they also exposed how little had previously been known. What divers discovered was not the full wall, but merely another layer beneath the familiar surface zone. Far deeper sections remained inaccessible until the emergence of closed-circuit rebreather technology. During the 1990s, increasingly sophisticated systems allowed technical divers to recycle breathing gases and manage decompression more efficiently. Descents approaching 150 meters became possible under carefully controlled conditions. Entire portions of the mesophotic zone, long hidden between shallow reefs and the deep ocean, entered direct human observation for the first time. Scientists encountered unfamiliar coral communities, unusual fish assemblages, and ecological relationships that challenged assumptions built solely upon studies of shallow water environments. Yet even these achievements represented only a fraction of the wall's true depth. The deeper sections required machines rather than divers. Remotely operated vehicles, equipped with cameras, lights, robotic arms, and scientific instruments, began systematic exploration during the 1980s. Freed from the physiological limitations imposed by pressure and decompression, these vehicles descended into regions that no human could safely visit. They returned with images of communities living hundreds and sometimes thousands of meters below the surface, expanding scientific understanding with every expedition. Among the most influential efforts were surveys conducted by the Pisces submersibles operated through the Hawaii Undersea Research Laboratory. These missions explored Hawaiian island walls below 200 meters and documented organisms whose known ranges required revision. Species believed restricted to one environment appeared in another. Communities once assumed rare proved unexpectedly common. The wall repeatedly demonstrated that it was more complex than existing models predicted. As technology improved, researchers discovered that even cameras could influence behavior. Many predators avoided the noise and lights associated with survey vehicles. To overcome this limitation, scientists deployed baited remote underwater video systems. Cameras attached to bait stations were left undisturbed for hours along wall faces, recording activity without the constant presence of operators. These systems revealed predator assemblages and feeding behaviors that earlier surveys had missed entirely. The next breakthrough arrived not through sight but through genetics. Environmental DNA analysis allowed researchers to collect seawater samples and identify species from microscopic traces of genetic material suspended within the water column. Organisms no camera had ever recorded could be detected through the biological signatures they left behind. Entire components of wall ecosystems became visible without direct observation. At the same time, multi-beam sonar transformed understanding of the wall's physical structure. Detailed bathymetric mapping revealed vast overhangs, enormous vertical relief, and cave networks extending deep into geological foundations. Features stretching hundreds of meters remained invisible even to explorers moving directly beside them. With each technological advance, the island wall became less mysterious, yet the expanding picture consistently revealed something unexpected. The deeper humanity looked, the larger and more intricate the hidden world appeared to be. Certain places in the ocean become more than locations. Through decades of observation, they become laboratories written directly into the landscape, places where the sea repeatedly reveals patterns that could not be seen anywhere else. Island walls have produced many discoveries, yet a small number of them have contributed so profoundly to scientific understanding that their names now echo throughout marine biology. Their importance emerged gradually, not from a single dramatic revelation, but from years of evidence accumulating layer by layer until entire assumptions about ocean life had to be reconsidered. Among the most influential is the Bahamas wall system surrounding the deep basin known as the tongue of the ocean. From above, it appears as a narrow corridor of dark water enclosed by islands. Beneath the surface, however, it provided one of the most valuable natural research environments ever available to shark scientists. During the 1960s and 1970s, researchers used this setting to conduct controlled studies of shark behavior and sensory perception. The steep geometry of the surrounding walls created conditions where animal movement could be observed with unusual precision. Experiments carried out there established foundational understanding of electroreception and chemoreception, revealing how sharks detect faint electrical signals and chemical traces in the water. Much of what is now considered basic shark biology traces its roots to work conducted along those walls. Far to the west, the Cayman Island walls revealed another hidden dimension of marine life. For generations, many deep reef fish appeared mysterious in their reproductive behavior. Scientists knew populations persisted, yet the locations and timing of spawning remained largely unknown. Careful observation along the Cayman walls eventually documented immense deep-water spawning aggregations gathering at specific sections of the wall face. These events demonstrated that reproduction was not randomly distributed throughout the habitat. Instead, geography itself shaped the process. Particular ledges, contours, and current conditions became recurring destinations where thousands of fish assembled. The discovery transformed understanding of reef fish ecology and highlighted the importance of protecting specific physical locations rather than treating entire habitats as uniform environments. Across the Pacific, the walls surrounding Cocos Island contributed another chapter. Strong currents sweep continuously across this isolated island, carrying nutrients and attracting vast concentrations of marine life. Here, researchers observed schools of hammerhead sharks gathering in numbers rarely seen elsewhere. For many years, the purpose of these aggregations remained uncertain. Continued study revealed a connection to thermal structure along the wall face. Rather than gathering randomly in open water, the sharks concentrated near thermocline boundaries, where water masses of different temperatures met. The wall created a framework that organized these thermal layers, helping explain a behavior that had puzzled scientists for decades. What appeared at first to be a simple congregation of animals proved linked to the physical architecture of the environment itself. Another turning point emerged from the island walls of Papua New Guinea. During surveys conducted throughout the 1980s, coral biologist J.E.N. Veron encountered coral communities of extraordinary richness. Species diversity exceeded expectations again and again. Each survey expanded the growing realization that this region represented something globally exceptional. The evidence eventually helped establish the Coral Triangle as the center of marine biodiversity on Earth. That designation reshaped conservation priorities, redirected funding, and influenced international policy. The walls themselves became part of the scientific foundation, supporting one of the most important marine conservation concepts of the modern era. Near the outer edge of Australia's Great Barrier Reef, Lizard Island added a different kind of contribution. Beginning in the early 1970s, researchers established one of the longest continuous marine monitoring programs ever maintained. Year after year, observations accumulated. Individual changes appeared subtle, sometimes almost imperceptible. Yet across 5 decades, the record revealed shifts in community structure, species abundance, recruitment patterns, and environmental conditions that would have remained invisible within shorter studies. The wall became a living archive of ecological time, demonstrating that understanding the ocean often depends not only on where scientists look, but on how patiently they continue watching. Even in places studied for generations, there remain observations that refuse to settle into explanation. Data accumulates, instruments improve, and theories become more sophisticated, yet certain events persist at the edges of understanding. Island walls, despite the attention they have received from biologists, oceanographers, and explorers, continue to produce records that fit no established pattern. These are not legends or unverified stories. They are documented observations preserved in scientific reports and survey records, acknowledged as real while their causes remain uncertain. Among the most puzzling are mass appearances of deep-water squid near the upper reaches of island wall systems. On several occasions in the Pacific, Researchers and local observers have recorded thousands of squid emerging from depths where they normally spend their lives and gathering near the surface interface adjacent to a wall. The events occur suddenly and often end just as abruptly. No consistent trigger has been identified. Temperature records, current measurements, lunar cycles, and chemical sampling have all been examined, yet none provide a universal explanation. The animals appear, concentrate in remarkable numbers, and then vanish back into the larger mystery of the deep. Elsewhere, long-monitored wall communities have displayed a different form of unpredictability. At several Caribbean sites, assemblages of fish that remained stable for years have disappeared within days. Species that occupied the same ledges, feeding grounds, and shelter sites through season after season suddenly abandoned entire sections of wall. Researchers searched for disease outbreaks, predator influxes, changes in current structure, and shifts in water chemistry. In some cases, no measurable alteration accompanied the departure. Months later, the same species returned, repopulating the area as though nothing had happened. The wall resumed its familiar rhythm, while the interruption remained unexplained. Farther west, the Mariana Island walls have generated acoustic recordings that continue to challenge classification. Hydrophones deployed along the slopes detected low-frequency tonal calls between roughly 30 and 60 Hz. The sounds possessed characteristics associated with biological sources rather than geological activity or mechanical interference. Yet comparisons against known marine mammals, fishes, and invertebrates failed to produce a match. The recordings have been cataloged and analyzed repeatedly. They remain present in the literature as recognized signals without a confirmed author, drifting through the scientific record like distant voices whose source remains hidden beyond observation. Light itself contributes, another mystery. Technical divers working along several island walls have documented episodes in which large sections of the surrounding water column began pulsing with coordinated bioluminescence. Unlike the brief flashes commonly produced by individual organisms, these events involved vast volumes of water illuminating in synchronized waves that persisted for minutes. Video records confirm the phenomenon, yet identifying the mechanism has proven difficult. A single species acting alone cannot easily account for the scale. The possibility of multiple organisms responding simultaneously to a shared environmental cue remains under investigation, though no definitive explanation has emerged. Perhaps the most enduring puzzle involves navigation. Across decades of tagging studies, researchers have documented sharks, rays, and other large pelagic animals returning to remarkably specific sections of island walls. Individual animals have been recorded appearing at the same locations year after year, despite traveling across immense stretches of ocean in the intervening periods. Chemical gradients, magnetic fields, acoustic cues, and inherited migratory behavior all appear to contribute in some measure, yet the precision often exceeds what existing models comfortably predict. A particular bend in a wall, a specific promontory, a narrow corridor shaped by geology millions of years ago, somehow becomes a destination remembered across years and across oceans. These anomalies do not undermine scientific understanding. They reveal its living edges. Along island walls, the known and the unknown continue to exist side by side, separated by surprisingly little distance. Each survey adds clarity, yet each answer occasionally exposes another layer of complexity beneath it. The wall remains a place where observation often arrives before explanation, where evidence waits patiently for the ideas capable of fully interpreting it. Long before changes become obvious across entire oceans, they often appear first in places where many forces converge at once. Island walls occupy exactly such a position. Stretching from warm surface waters into deep darkness, they connect biological communities, chemical gradients, and physical processes that elsewhere remain separated by immense distances. Because so many layers of the ocean intersect along a single vertical landscape, even subtle shifts leave traces. The wall responds, and in doing so, it quietly records the direction in which the broader ocean is moving. One of the clearest signals comes from coral bleaching. For decades, scientists understood bleaching primarily as a phenomenon of shallow reefs where prolonged heat stress causes coral to expel the microscopic algae living within its tissues. Without those algae, coral loses much of its color and much of the energy source that sustains it. Along island walls, however, researchers have begun documenting bleaching events far deeper than expected. Communities once considered insulated from surface warming are now showing signs of stress. Heat is not remaining confined to the upper layers of the sea. It is penetrating farther downward, altering environments that earlier models treated as comparatively stable. This realization has transformed understanding of mesophotic reefs, those dimly lit coral communities occupying the middle depths of island walls. For years, these reefs were viewed as potential refuges. As shallow habitats experienced increasing thermal stress, conservation planners hoped deeper communities might survive and eventually help replenish damaged areas above. Yet surveys have revealed bleaching within these deeper ecosystems as well, occurring at depths once believed secure. The assumption that a protected sanctuary existed farther down the wall has become increasingly difficult to maintain. The same environmental pressures influencing surface communities are reaching into regions previously thought sheltered from their effects. The changes are not limited to coral. Across many island walls, species distributions are shifting in measurable ways. Tropical organisms are appearing farther from the equator, establishing themselves in wall environments that historically remained outside their preferred range. At the same time, species adapted to cooler conditions are retreating downward, following the temperatures to which their physiology is best suited. Because island walls compress such a large depth range into a relatively small horizontal space, these movements become visible with unusual clarity. The wall functions almost like a vertical map, displaying biological responses to changing climate in real time. Chemistry adds another layer to the story. As increasing amounts of carbon dioxide dissolve into seawater, ocean acidity gradually rises. The shift is subtle when measured over short periods, yet its consequences accumulate. Many organisms living on island walls depend upon calcium carbonate to construct skeletons, branches, and supporting frameworks. Deep water corals, gorgonians, and countless smaller builders invest energy into structures that become the architecture of the ecosystem itself. Researchers have documented signs that some of these structures are becoming thinner and more fragile. Growth continues, but the balance between construction and dissolution is changing. Beneath these observations lies an especially significant boundary known as the aragonite saturation horizon. Above it, calcium carbonate structures can form and persist more easily. Below it, dissolution becomes increasingly favorable. Measurements indicate that this horizon is rising through parts of the ocean. As it moves upward, communities occupying deeper portions of island walls encounter conditions less supportive of the very materials from which they are built. Pressure emerges from below, while warming advances from above, narrowing the environmental space available to many long-established organisms. For this reason, Island walls have become something more than remarkable habitats. They have become observatories. Their extraordinary sensitivity allows scientists to detect biological, chemical, and physical change while those changes are still unfolding. Each current shift, each alteration in species distribution, each modification in water chemistry becomes part of a larger pattern. The wall gathers these signals across its entire depth, preserving them in living communities that respond almost immediately to changing conditions. In that sense, island walls offer one of the clearest views available into the future trajectory of the global ocean, revealing transformations while they are still in progress rather than after they have fully arrived. Consider a map whose outlines are drawn with confidence while the vast majority of its interior remains blank. From a distance, it appears complete. Only when examined closely does the scale of the unknown become visible. Island walls occupy a similar place in modern science. They have been photographed, sampled, measured, and discussed for decades. Yet the deeper researchers look, the more apparent it becomes that understanding remains concentrated within a remarkably small fraction of the habitat itself. One reason is scale. No definitive calculation exists for the total length of island wall habitat across the world's oceans. The challenge is greater than it first appears. These environments are not simple coastlines that can be traced across a map. They descend vertically, branch into ledges and overhangs, fold around volcanic islands, disappear into cave systems, and continue into depths that remain difficult to survey. Estimates range from hundreds of thousands to more than 1 million kilometers of vertical habitat. Regardless of which estimate proves most accurate, the conclusion remains the same. The overwhelming majority has never been examined directly. Even within the portions known to science, observation becomes increasingly sparse with depth. Above the shallower zones, divers and researchers have accumulated decades of records. Beyond roughly 150 meters, however, the picture changes dramatically. Systematic surveys become rare. Instruments are expensive, deployments are limited, and exploration often consists of brief transects crossing immense landscapes. When scientists describe a wall as surveyed, the phrase can conceal how little of that environment was actually observed. A handful of camera passes may represent knowledge of an area extending far beyond the limits of direct inspection. Yet perhaps the most remarkable pattern is what those limited surveys continue to reveal. Across decades of exploration, deep wall expeditions have repeatedly encountered organisms unknown to science. New fish, corals, crustaceans, worms, sponges, and microbial communities continue to emerge from habitats that had never previously been sampled. The trend has persisted with striking consistency. Rather than approaching a point where discoveries become rare, researchers continue finding evidence that the catalog remains profoundly incomplete. Each descent suggests not that the inventory is nearly finished, but that it has barely begun. The implication reaches beyond simple numbers. Every undiscovered species represents an entire evolutionary history unfolding without human awareness. Generation after generation has lived within the architecture of the wall, adapting to currents, predators, food sources, and environmental pressures across spans of time measured in thousands or millions of years. Complex relationships have formed. Specialized behaviors have emerged. Entire ecological networks have persisted unseen. Their existence has never depended upon observation. They have continued whether anyone watched or not. This perspective introduces a rare kind of humility into scientific exploration. Modern technology can map distant planets, sequence genomes, and monitor weather systems across continents. Yet along island walls, vast communities remain effectively invisible. The limitation is not a lack of curiosity or effort, it is a reflection of the extraordinary complexity of the environment itself. The wall compresses immense depth, countless microhabitats, and biological interactions into a space that remains difficult to access even with the most advanced equipment available. What makes this reality especially striking is its immediacy. The unknown is not confined to ancient history or remote geological epochs. It exists in the present moment. Somewhere along island walls scattered across the Pacific, Atlantic, Indian, and Southern Oceans, organisms are feeding, reproducing, migrating, competing, and communicating beyond the reach of current observation. Currents continue shaping habitats that no camera has recorded. Predators move through darkness where no instrument is waiting. Entire communities carry on their existence without witness. After everything science has learned from island walls, the most accurate conclusion may also be the simplest. These environments have revealed enough to transform understanding of the ocean, yet they continue to demonstrate how much remains beyond the edge of knowledge. The wall stands as a reminder that discovery is not approaching an ending. In many ways, it is still at the beginning. There is a moment in any long exploration when the details begin to gather into a larger pattern. Individual species, current systems, chemical exchanges, migrations, and geological structures cease to feel like separate subjects. Together they reveal something broader about the nature of life itself. Seen from this wider perspective, island walls become more than marine habitats. They become a demonstration of a principle that appears again and again throughout the natural world, expressed here with unusual clarity and scale. Life is often imagined as seeking stability, simplicity, and ease. Yet the evidence preserved along island walls points in another direction. The greatest concentrations of diversity are rarely found in the most uniform environments. Instead, they emerge where different conditions meet. The richest stories of biology tend to unfold at boundaries. Along the edge of forests, species from separate habitats encounter one another. Along river margins, freshwater and terrestrial ecosystems intermingle. In estuaries, saltwater and freshwater overlap, creating communities that neither environment could support alone. Nature repeatedly concentrates complexity at transitions. Island walls represent perhaps the most dramatic transition available anywhere in the ocean. Across a relatively small horizontal distance, sunlight fades into darkness. Warm water gives way to cold, shallow conditions, descend toward immense pressure, and surface communities exist alongside organisms whose evolutionary histories unfolded far below. These worlds remain distinct, yet they touch continuously along the wall face. The result is not a narrowing of possibilities, but an expansion of them. Each gradient creates opportunities for specialization, adaptation, and interaction that would not exist within a more uniform setting. The creatures living there illustrate this principle in countless ways. Some exploit narrow bands of temperature found only at specific depths. Others depend upon the movement of nutrients between layers of the ocean. Predators encounter prey originating from different ecological zones. Corals, sponges, fishes, and microorganisms occupy niches defined not by a single condition, but by the intersection of many. Diversity emerges not despite the complexity of the environment, but because of it. The wall transforms contrast into opportunity. This pattern extends across time as well as space. Ancient coral colonies anchored to deeper sections of island walls, have persisted for periods spanning many human civilizations. Some began their lives long before events that dominate written history. Yet describing these organisms merely as survivors misses something important. Survival suggests endurance against an environment. These corals are not enduring their habitat. They belong to it. Their extraordinary longevity reflects a place capable of supporting continuity across centuries and millennia. Part of that continuity arises from the distinctive stability of deeper wall environments. Surface ecosystems experience storms, seasonal fluctuations, and abrupt changes driven by weather. Conditions farther down evolve more gradually. Temperatures remain comparatively constant. Light changes little because little light arrives at all. Biological communities persist within rhythms measured across longer spans of time. This stability has allowed evolutionary processes to unfold with remarkable continuity, preserving lineages and ecological relationships that might struggle to persist in more variable settings. What emerges from all of this is a subtle lesson about where biological richness originates. The most interesting processes are not always found at the extremes themselves. Neither the brightest shallows nor the deepest abyss tells the complete story. Instead, much of the ocean's creativity appears concentrated in the spaces where those extremes interact. Boundaries become engines of innovation. Gradients become opportunities. Differences become sources of connection rather than separation. Island walls make this visible. They reveal an ocean organized not only by distinct environments, but by the exchanges between them. They show that life often flourishes most vigorously where conditions overlap, where contrasts meet, and where separate worlds remain close enough to influence one another. Along these vertical landscapes, the fundamental logic of biology becomes difficult to miss. Complexity grows from contact, diversity emerges from transition, and the meeting place between worlds becomes a world of its own. A narrow beam moving through darkness reveals only a small portion of what surrounds it. Everything beyond the light remains present yet unseen, continuing its slow existence outside immediate perception. Along the deeper reaches of an island wall, this simple reality becomes impossible to ignore. The environment is not empty, it is densely inhabited. Yet, so much of it exists beyond the limits of human senses that the experience creates an unusual impression, one often described as silence. At depths of roughly 200 meters, where sunlight has long since faded from practical influence, the familiar soundtrack of the surface world no longer dominates awareness. Waves no longer announce themselves. Wind no longer presses against the water above. The noise of daily human activity feels impossibly distant. Instruments can detect vibrations, biological signals, and the movement of water, but the human mind often interprets the overall experience as quiet because the patterns are so different from those that shaped our species throughout its history. The silence of the deep wall is therefore not an absence, it is a different kind of presence. A slow current slides along stone that has stood in place for geological ages. Tiny organisms feed across mineral surfaces. Filter feeders process passing water one particle at a time. Somewhere far below, a sperm whale releases a sequence of clicks that travels through the water with extraordinary efficiency. The sound may originate hundreds of meters away, yet it arrives with startling clarity before fading once more into the surrounding stillness. Between such moments stretches a vast acoustic landscape shaped by patience rather than urgency. Many creatures living along these deeper sections inhabit a soundscape fundamentally unlike the one known near the surface. Large portions of their lives unfold beyond the reach of engine noise, harbor traffic, and the persistent mechanical rhythms associated with human civilization. Although modern ocean noise can penetrate considerable distances, numerous deep wall habitats remain comparatively insulated, preserving conditions closer to those that existed before industrial activity expanded across the world's oceans. Generations of organisms have evolved within this quieter framework, responding to biological cues carried through water rather than the constant mechanical signatures familiar to human ears. Researchers descending into these environments often report that the experience affects them in ways difficult to quantify. The physical conditions alone do not fully explain the reaction. Depth, darkness, and pressure contribute to the sensation, yet there is something else at work. Unlike the open abyss, which can feel boundless and abstract, an island wall provides orientation. It possesses shape, texture, and continuity. The eye can follow its contours upward toward distant brightness or downward into fading obscurity. The structure creates a sense of place within an environment that might otherwise seem immeasurable. That geography allows the mind to engage with scale differently. Distance becomes visible rather than theoretical. Time also acquires a new character. Organisms attached to the wall often grow at rates so slow that a single human lifetime represents only a fraction of their existence. A deep water coral colony, several meters across, may have begun growing centuries before many modern nations existed. Some of the oldest specimens found on island walls have persisted for roughly 2,000 years, adding tiny increments of living structure year after year, while empires rose, expanded, fractured, and disappeared far above. Seen directly, such longevity ceases to be an abstract figure recorded in scientific literature. It becomes a living presence, occupying the same space as the observer. The colony continues its patient growth exactly as it did before ships crossed oceans under steam power, before global communication networks connected continents, before countless generations experienced their brief passage across the surface world. The wall holds these lives within its architecture, preserving a continuity rarely encountered elsewhere. In that quiet environment, surrounded by organisms whose histories stretch beyond ordinary human reference points, the silence begins to feel less like emptiness and more like duration itself, a living reminder that vast portions of Earth's story continue unfolding slowly, steadily, and almost entirely beyond our notice. The most revealing shift begins with a simple change in perspective. Imagine standing on a shoreline, looking out across calm water toward an island that appears small, familiar, and entirely knowable. The scene seems contained within ordinary human experience. Boats move across the surface. Sunlight reflects from gentle waves. Life unfolds according to scales that feel comfortable and understandable. Yet attached to that same island, hidden beyond the visible edge of the reef, a vertical world descends into depths commonly associated with the most remote regions of the planet. For generations, the deep sea has occupied a particular place in the human imagination. It has been presented as the final frontier, a realm defined by distance, darkness, and inaccessibility. Its mysteries feel immense because they exist far beyond normal reach. The abyss lies below layers of water that separate human life from the creatures and processes inhabiting it. That separation creates a psychological boundary as powerful as any physical one. The unknown remains comfortably distant. Island walls disrupt that idea completely. The extraordinary depths associated with the deep ocean do not always begin far offshore. In many places, they begin astonishingly close to the surface. A swimmer floating above clear tropical water may be suspended over a geological drop that continues downward for hundreds or even thousands of meters. The transition from familiar reef to deep ocean occurs not across vast horizontal distances, but across a vertical face that compresses entire worlds into a single structure. This changes the question the ocean asks. A six-gill shark ascending along a wall after sunset is not emerging from some unreachable trench hidden beyond the horizon. It is moving through water beneath vessels whose occupants may be asleep only a short distance above. Ancient coral colonies that have persisted for roughly 2 millennia are not confined to remote abysses beyond the reach of observation. They continue their patient growth beneath reefs, visited by divers every day, existing just beyond the depth where most human exploration naturally ends. The same pattern repeats throughout the story of island walls. Unidentified acoustic signals originate not from isolated corners of the planet, but from environments attached directly to inhabited islands. Bioluminescent events, still lacking complete explanation, occur beside coastlines celebrated for their beauty. Species found nowhere else on Earth survive within systems connected to beaches, harbors, fishing grounds, and tourist destinations. The realization is subtle but profound. The ocean's most extreme biology is not necessarily concentrated in the deepest trenches or the most inaccessible basins. Again and again, island walls demonstrate that extraordinary complexity emerges where radically different environments meet. Surface and abyss, warm and cold, light and darkness, familiarity and mystery. The wall gathers these opposites into a single place and allows them to exist side by side. Seen from this perspective, the deep sea becomes less a distant destination and more a neighboring presence. Its creatures, processes, and unanswered questions are not separated from everyday human life by impossible distances. They exist directly beneath landscapes that appear on maps, postcards, and travel brochures. Entire biological communities unfold in quiet continuity below waters crossed by ferries and fishing boats. That recognition alters the emotional geometry of the ocean. The unknown no longer resides exclusively somewhere far away. It occupies the spaces immediately adjacent to the known. The boundary between ordinary experience and profound mystery becomes remarkably thin, sometimes no more than a descent along a submerged cliff. By revealing this proximity, island walls offer a different understanding of exploration itself. The deepest question is no longer centered on what may exist in some distant darkness beyond reach. Instead, attention turns toward the hidden worlds already surrounding us, present all along, continuing their ancient rhythms beneath the surface, waiting not to be created or discovered, but simply noticed. The descent ends where it began, suspended beside a vertical face that vanishes into darkness. Yet the darkness no longer carries the same meaning. Earlier, it seemed empty, a blank space beneath the known world, a region defined primarily by absence. Now it feels crowded with history, structure, and life. The blackness remains, but it has acquired depth in a different sense. It contains ancient colonies anchored to stone, predators tracing routes older than memory, hidden chambers carved through geological ages, and communities that continue their quiet existence beyond the reach of ordinary observation. What once appeared to be nothing has revealed itself as an intricate geography. The wall possesses architecture, it possesses chemistry, it possesses currents that connect surface waters to the deep ocean below. It possesses signals carried through sound and scent. It possesses organisms found nowhere else on Earth. Across its face grow corals, whose lifespans stretch beyond the rise and fall of kingdoms. Along its slopes move animals that navigate darkness with a precision science is still attempting to understand. Within its caves survive lineages, isolated for immense spans of time, continuing evolutionary stories that unfolded unnoticed while human history followed its own course above. The remarkable truth is that none of these things arrived recently. They were already there when the earliest ships crossed unfamiliar seas. They were there when ancient ports flourished along distant coastlines. They were there while empires expanded, fragmented, and disappeared. Throughout every chapter of civilization, island walls remained fixed at the boundaries between light and darkness, sustaining communities whose existence was completely independent of human awareness. A colony of black coral growing deep beneath a Caribbean island may have begun its life long before the first European vessel entered those waters. Year after year, it added tiny increments to its structure, recording chemical traces of the surrounding ocean in its skeleton. It continued through centuries of storms, migrations, exploration, industry, and technological transformation. While human societies accelerated through eras of extraordinary change, the coral maintained its slower rhythm, extending itself by mere fractions of growth each season in perpetual darkness. Even the ability to visit such places arrived astonishingly late. For most of human existence, these environments existed beyond direct reach. Entire generations lived and died without knowing what occupied the depths beneath familiar islands. Only within recent decades have technologies allowed consistent exploration of zones that had remained inaccessible despite their proximity. The wall did not change during that time. Human knowledge did. Seen from this perspective, the most significant lesson is not scientific. Although science has illuminated much, the lesson emerges from scale. Island walls reveal a continuity so vast that human history occupies only a narrow interval within it. Beneath the movements of nations and cultures lies another story unfolding according to different clocks, measured not in decades or centuries, but in ecological persistence, geological transformation, and evolutionary time. The recognition is quiet. It asks for no dramatic conclusion. It simply places humanity within a larger context. The ocean beneath islands existed before any chart recorded it. It shaped lives before language described them. It supported ecosystems before anyone imagined their existence. Long after current generations have passed into memory, those same processes may continue, currents flowing across stone, organisms feeding in darkness, ancient colonies, adding another small layer to structures already older than many civilizations. Somewhere along a wall face, at this very moment, beyond the reach of sunlight and beyond the attention of any observer, an unnamed organism continues its ordinary life. It moves, feeds, reproduces, and responds to conditions that have shaped its lineage for ages. It requires no witness. It follows patterns established long before human arrival. Perhaps that is what the wall ultimately asks of us. Not fear and not certainty, but perspective. A recognition that the living world is deeper, older, and more continuous than our experience alone can reveal, and that beneath the familiar surface of the planet, entire realms persist in patient silence, carrying on exactly as they always have.